Hello and welcome back to Intro to Cognitive Science. So, we have been covering ways that we have begun to use better understanding of the actual brain and the neurons involved in such a brain to make better sense of how a mind like a human's can come to, or rather come out of, that. And so we're going to continue exploring these questions of how neuroscience has sort of evolved along these lines. And today we're going to talk about the sort of a hallmark of a lot of neuroscience for several decades, which is trying to map processing in the brain. So, early neuroscience, as we have, discussed, a lot of times it was traumatic brain injury patients that would help us figure out the importance of one particular brain region to cognitive functions, right? And the experiments all required surgical interventions and especially animal research, as we've discussed. And that all worked, of course, and it's still important for understanding certain things about the brain today. We also looked at connectionist models, so how neurally inspired algorithms can connect to the things that we know about the brain and give us this sort of neural version of how computation could work in a mind. So now we're going to turn back to something that we discussed, a couple lectures ago, and talk more about neuroimaging techniques. So the dawn of these really, really more and more powerful neuroimaging techniques has allowed for this non-invasive, non-surgical, non-damaging look at the function of the brain, again, without having to disrupt the organism, the animal, the human, but just stick them in a tube for a while, right? And we'll do some high-powered energy transfer, and your brain waves will disrupt it, right? Well, that's a pretty coarse way to talk about it. We'll get into the details. But the nice thing about this is that by making it non-invasive, this allows us to look at how humans do kind of more interesting tasks. More complex things than just, say, a landmark study with a monkey trying to get kibble, right? Sorry, became muted. To break us into the topic, I'm going to let 2-Minute Neuroscience take over for a sec.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss neuroimaging. I will cover a few of the most common types of neuroimaging: CAT scans, MRI, PET scans, and fMRI. Some neuroimaging techniques allow us to see the structure of the brain, while others allow us to look at brain activity or function. Computerized tomography, also known as computerized axial tomography, is a type of structural neuroimaging. It is usually called a CAT or CT scan. It involves taking a series of X-ray images from various locations around the head. These images can then be combined to construct an image of the brain. The resolution of CT images is not that high, but they can visualize any major structural problems with the brain, like a tumor. Magnetic resonance imaging, or MRI, involves applying a combination of magnetic fields and radiofrequency energy waves to the brain. Hydrogen atoms respond to the magnetic fields and radiofrequency pulses by emitting energy. The MRI machine receives this energy and can tell what part of the brain it came from. A computer can use that information to reconstruct an image of the brain that has high spatial resolution. Positron emission tomography, or PET scanning, is a way of imaging brain function. To do a PET scan, a patient is injected with a radioactive substance that emits positrons, which then emit gamma rays when they collide with electrons in brain tissue. These gamma rays are detected by the PET scanner. Because the radioactive substance was injected into the bloodstream, what the PET scanner is detecting is the movement of blood throughout the brain. Blood flow to an area of the brain increases when that area is active, so PET scanning creates an image that highlights the areas of the brain that are being used most while the person is in the scanner. Functional MRI, or fMRI, uses a similar approach to MRI but focuses on the different responses oxygenated and unoxygenated blood make to magnetic fields and radiofrequency energy. fMRI uses what is called blood oxygen level dependent contrast, or BOLD, to identify changes in blood flow in the brain, and thus to identify areas of the brain that are most active. fMRI allows one to image brain function without having to inject anything, and it provides high-resolution MRI images at the same time as it provides a functional image.
Okay, that was fairly dense. In fact, I may stop using this particular video, but what I want you to get from this is that there have been a lot of different techniques that evolved that allow for mapping different things. And what we're getting to is a connection between the spatial understanding of the brain, which we were able to achieve with CAT scans and normal MRI, and a functional understanding of the brain. So this is a strong distinction between what's available. So again, structural information we can get from CAT scans and MRI scans. So we get the locations and shapes of the brain regions, right? Whereas functional information we can get from things like PET scans, which are, again, they are measuring some amount of the blood flow in the brain using an injected dye. We'll talk about that. But it gives you the activity, right? But with a little bit poorer spatial clarity. Better is the sort of the one that, just flashing forward, the one that has become the most common because it's so useful in both medical settings and scientific settings, is functional MRI. So the ability to detect the blood flow and the spatial information quite well together. So what can we do with this? So this is just setting the stage. These are the tools that have evolved. And the question is, how can we use this to start asking deeper questions about how does the brain do what it does? What are some of the interesting functions that we can tackle? Well, today we're going to talk about language again, as we like to. If you recall, Sherdlu showed us some of the functions you'd probably need to understand language processing, right? It had various modules, you could call them, different little elements that were doing the processing in parallel together. And a few of them were kind of critical, right? We needed some kind of semantic processing, some kind of syntactic processing of the language coming in to make sense of when I tell it, hey, put the blue triangle on the green box. It's like, yeah, okay, I can do that, right? How did it do that? Well, that's not necessarily germane to how our brain does it. But the question is, how does our brain do it? So the big question of today's investigation is, what's in a word, right? There's information involved in a single word, right? But there's a lot of different kinds you might not have thought of. So there's how it looks, the exact shape and layout of the words in front of your eyes, right? There's how it sounds. There's the auditory information. When I say sounds, that is a set of vibrations moving through a microphone into a computer and then decoded on your end in the video and out through the magnets into your ears. That's a lot of stuff, but it's information. It's the auditory shape of the word mapped together with the look, the visual shape of the word. And there's also ultimately, what does the word mean? What is the actual meaning of the word? So, as discussed last time, there's this push forward into thinking about, how does this information move around? How does it flow in the brain, right? And there's two different models about how we understand words, pick out the features of words and make use of them. There is the serial processing model, which says that, hey, information comes in through the earwaves or the eye holes, very technical terms. Ear holes or eye holes, regardless of where it comes in, and it first gets processed as visual appearance, then it might go through sound, either way. So the visual pattern must get transformed into a sound, and then that sound gets transformed into meaning, right? And so if you're thinking, well, that doesn't make much sense, the idea here is that all information is kind of flowing along the same tracks. This is the same kind of stuff that we were talking about last week, or last lecture, and trying to understand, okay, is everything moving in a serial kind of lockstep pattern? So does the visual word actually kind of hit an auditory processor, or does it jump straight to meaning? Whereas the parallel processing model is saying, hey, that we think it's probably more like a Sherdlu, right? Information's moving forward, and it's processed simultaneously in multiple locations, but those will all kind of jump to meaning eventually. They don't have to stop along these tracks. So that might be familiar from connectionist network ideas, right? So the question is there evidence in the brain for either of these patterns, right? So if I am trying to chart what's happening in your brain when you're doing lexical processing, you're coming to understand a word, can I show that? Up until this point, the answer was firmly no. But now that these tools are arriving, Peterson can come in and say, let's try. So, 1988, Peterson's and company are interested in how words are processed. So, the same question. And they designed, I really like this study a lot. It's a contrastive study to examine how does that information flow in the brain. And how much overlap is there between both seeing or hearing a word in terms of what parts of the brain are active, and simply just repeating a word or understanding a word, demonstrating that you understand the word. Let's look. What did they use? PET scans. So while I did just talk up fMRI as a stronger option, PET scans still give you some spatial information. They allow all of these involve you go and lay down in a big tube, right? And you just kind of stay still as much as you can, and you do some task, right? In this case, in PET scans, they involve. So a PET scanner will have to shoot positrons through you. I know that sounds terrifying in some ways, but normally positrons don't interact with us, humans, or any physical things. What they do is they inject a little radioactive substance into your body, into your blood, that now you will interact with those positrons. Again, oh my gosh, that sounds terrifying, but ultimately, all of these things are pretty inert. I can't think of a single case of someone developing a disease from a PET scan. So, despite the high sci-fi nature of it, all it's saying is, I'm going to put this weird water in you, and you're going to, and that's going to show me where the blood is in your body, and specifically in your head, so that your blood flow is going to show up on X-rays. The positrons involved will interact with just your the weird water, right? The radioactive substance, and not everything else. Okay, that was perhaps too long an explanation, but I hope you understand now how this is kind of working. And what we can do with this is we take these pictures, we say, hey, when I'm having you solve this particular little task, little mental exercise using words, what does your brain activity look like? So here's the study. Overall design. So it's called a contrastive study because they take a baseline first, and they're going to keep layering more and more complex versions of the task on top. So let's look at what is baseline in this case. Well, the baseline condition is stare at a fixation cross. That's nothing. That's just a little plus sign in your field of vision. And the neural processes involved here, hopefully, would be a baseline resting state, right? So this is just the neural activity we expect is just kind of whatever your neurons are doing normally, right? Thinking about breakfast or plans for dinner. It's all food with humans in it. And that's it, right? So the stimulus they would see would be something like a plus sign, right? In a dark field. The response requested is nothing. So what they would do is they would record the neural activity using the PET scan during this baseline, and now when we go to something more complex, we can subtract all the baseline neural activity off of the resulting image. You'll see what I mean. So next would be a passive vision condition, asking the question. So the task is to just view some words as they appear. And the question that what this is trying to locate, what this is trying to find in the brain, is visual processing at a very basic level. Just seeing and processing something visually. So in this case, the stimulus you see would be bird, but the response is there's still no response. You're not being asked to do anything with the information. Just look at it. This presumably would not recruit other parts of your brain that would be involved in doing something interesting with the word. It's just the word. Okay, cool. So now, if I have a scan of your brain when you're processing the word bird, and a scan of your brain when you're kind of doing nothing, I can subtract the two. I can, as always, it's just math, right? So it's just an activation value at a specific location. Subtract that off, we get, hopefully, where is the differential of what happens in your brain to process bird. We can do the same with audition or auditory processing. In this case, they would just hear words. And again, trying to figure out auditory processing. Where is that located? Subtract baseline from this, and you would get where the auditory processing is. So their stimulus in this case is not going to be a word or anything visual. It'll just be the sound bird, right? The word bird. Yeah, of course. Still no response, right? So we're still just seeing what is the processing going on for each one of these. Okay, now we start to continue to get more complex. Now we introduce the speaking condition. Okay, so in this case, the task is speak the words. Okay, so now we're trying to see what in addition is required for speech production. Not just taking in the word and not just like processing it, but processing it with the intention of using it. And in this case, they used a visual stimulus again, but the outcome, this time, they have a response. They want you to make a response. So now, okay, what are we doing here? We're not going to actually subtract baseline off of this condition. In the speaking condition, we're going to subtract off the vision condition because we are having them view the word bird and all plus all the resting state stuff. And now we're doing that again, but we're also having them produce the word bird. You can subtract off vision and see what is involved in speech production. Finally, we have the most complex one, which is verb generation. So this one, your task is to produce an appropriate verb, right? Something that this noun does, right? So the neural process involved here would be much more like semantic networking, right? So you're not just processing the word and spitting it back out. You're processing it, and then you're consulting your knowledge, right? What the heck does a bird do? Well, it probably flies. So in this case, we'd see the word bird again, and we would say the response would be fly. Okay, that's a lot. There's a lot going on here, and that's great. You know, we prefer that sometimes. But I hope you're getting the picture that this is a clever way to say, okay, I think I can locate where various kind of fundamental processes in this language processing task live in the brain. because I have a pretty decent spatial understanding of where these things are going to happen, and I can get an overall picture by isolating them. Okay, so the results. First, basic success. There are very different patterns of activation between the tasks. Fantastic. So we can see, I'll zoom this in. When you're passively viewing words, it's mostly occipital cortex stuff, back of the head, eyes, the visual stuff. That makes sense. When you're listening to words, it's much more the temporal lobe is active, right? So the middle side brain. When you are forced to speak words, there's a little bit of temporal activity that's unique there, but also kind of like motor strip stuff going on. That's the region of your brain that we've isolated to, more about producing actions. And then generating verbs, there's a whole lot of other structures involved, things up in the forebrain, all kinds of stuff, and a little bit of temporal and memory things. So I'm kind of layering in my own interpretation of these charts, but and kind of spoiling that, I'm flashing forward to years of our already understanding these things. But what's exciting is you can see this, right? They have isolated different processes to these locations. This doesn't mean that's exclusively where these things live, because there's a lot of noise in all these data sets, but it's exciting. You're starting to see how we've been able to chart where things live in the brain. So ultimately, there's a bunch of evidence here against the serial processing model, which you should have expected, I think, right? I didn't do a good job of setting it up as a potential thorough alternative, but back then it really could have been, because there's no reason we should have expected everything. There's no telling how it worked until we actually found it. So, okay. But the specific evidence against the serial processing model are things like, in the speaking condition, you say words that are presented visually, and there's no auditory processing at all. Now remember, in the serial processing expectation was, it's going to have to hit all of these places, right? The information is going to have to come in and hit everywhere in the chain, because even though it might not be necessary to go there, it still is going to have to go through there, right? The answer is no. It can totally skip all auditory processing, even though you're making people say the word, right? So this skips the whole idea of like things moving from one station to the next. There's no need to sort of like recode the visual version into some auditory form so that the auditory form can then be spit back out in your speech production, right? What's really more likely here is that there's these distinct paths from both visual and auditory areas into the speech production area. Because why not? That's something that we need. That's something that we vastly benefit from, and wasting the energy. If you learn anything else from this class, you should probably learn that the brain is pretty lazy. It doesn't really want to solve a million different things. It wants to do things the simplest way it possibly can. That doesn't mean it always succeeds to, but that's one of its goals. So when you think about brains, think lazy neural wiring. And I kind of mean that. So, okay. So another interesting element here that came out against the serial processing model is the idea that the semantic areas of the brain were only ever used in the semantic task, right? There was no overlap at all. You didn't end up accidentally triggering your semantic lookups when you weren't being asked to do anything with the word, right? You were just being asked to spit it out. So speech production doesn't need semantic activation. So again, that's one of the many stops on the train, on the neural train, right? So what we can find is, if we look at this model that Peterson brought to the table here, it's pretty nice actually. There's a lot in it, but we see he's broken the whole idea up into these distinct modules that only get used when they're needed, right? The visual processing might go and talk to the association, the semantic processing in the middle there, but it might just skip it and go straight to speech production. it only needs to go to speech. If all I need is speech production, then I'm not going to consult memory and semantic understanding. This is good. This is like good design compared to the sort of inefficiencies you would find in a very serial model. So understanding that these different areas can now kind of talk to each other independently of one another gives you a better understanding of the nature of the brain, and it certainly helped the field to do the same. So, to sum that up, to approach this attempt to map the different lexical processing elements of the brain, we're seeing that we can use brain imaging techniques to ask these really precise questions about the nature of like the flow of information in the brain. So, at a base level, which regions were used for which tasks, it was not exciting to me that the temporal lobe was being used for the auditory processing, right, in the findings here. But back then, that was new. These things were all becoming new and better understood, and in higher and higher resolution, higher and higher detail of what's used where. And it's also kind of helping with these higher order algorithmic questions of like, okay, if this region is responsible for this kind of processing, then when it becomes activated in this kind of task, that's interesting. That means that we're using spatial information, and it just helps to push and pull, right? And so when are things moving in parallel, and when might they be sort of bottlenecked when you need a certain kind of process before the rest can all move on? And in general, it's just, like I said, it's helping us. These kinds of things really help us make way better sense of the brain, and not just the minds that live on top of the brains, right? That's kind of a tricky way to put that. But within the brains, among the brains, emerged from the brains, right? There's a lot of different philosophical ways we can talk about that. But in general, this is great. I hope this illustrates how the brain imaging has really pushed us forward. We can also talk about a more recent study, sort of an interesting phenomenon, and this touches on that same thing I said before about models in science are primarily good if they can predict behavior, right? So, a good way to say that we can understand a system is being able to predict it. And Gallivan in 2011 was able to do that pretty quickly using brain data coming in. They could predict the motor actions that people took in this task before the participants actually performed them. Yes, you are predictable. Let's find out. So in this study, there were a handful of actions that people were supposed to take, right? And they were instructed to do them in response to certain scenarios, right? And the idea is there is this simple object, right? It's sort of a square with a smaller square kind of pushed on the top. Cube. 2 cubes, not squares. I swear I know geometry. So there were 3 kinds of things that they could do. There was grasp the bottom, grasp the top, and touch, right? So grasp the top is sort of finger and thumb are both involved, and it's sort of a fine motor thing. Grasp the bottom is a little bit broader action, still finger and thumb, and then touch is just the thumb on the side. And what we can see is using their techniques, fMRI, and a lot of data collection from a lot of different subregions, right? They've had, at this point in the development of neural research, there's a lot of very small regions available that people are pretty sure do certain things. Now, again, this is all, we're kind of shifting towards more network-based understandings of these things and not just this specific processor does 1x, right? But the idea here is that they have this wealth of data available, and using the activity from some of these regions, they're able to call out this person is about to do this action, right? And to give a little quick look at all of the ones available, again, you will not be responsible for these. This is just to show you. There's a load of different little areas. Even I don't know most of these, but like what they are specifically, they're known to do, right? But taking the activation patterns from all the different places, they build models. They build predictive models. And they say, okay, we expect that based on this pattern of activation, they are about to do this behavior. Ooh. So, and again, there's a lot of things here. We don't need to focus on all of them. But, so this is the idea. So again, to look at this really quick, the illuminator comes on to say, hey, here comes a part of this task. And they'll hear what they're supposed to do. And then they'll hear a beep that tells them when it's time to actually execute the thing. And the only data available to the models is this data in this time range between 12 and 16 seconds, which is about 4 seconds before the beep. The key thing here is that the model cannot see any activation of the brain during the instructions, during the illuminator. It can't even see it during the beep. Once the beep comes on, the model, it no longer knows what's going to happen. There's no understanding of what is about to happen. And by the time the actual execution occurs, around 22 seconds, then, the model's long gone, right? But here's the trick. You take that little set of data, and this is an abstraction, so it's multidimensional. It's not a single value, but this is just to help illustrate it. And these 3 different patterns that, again, come from many, many different observations across the brain, not just a single value, but the patterns represented here in this 4-second span, they're subtle, but they allow full prediction of what physical action is about to occur. So they're taking this little slice from just the brain activity with no understanding of the rest of the task and saying, I can tell what action is about to be executed. The idea being that there is this crucial motor planning step. After they've heard the instruction, but before they're instructed to do it, they've kind of primed their system to be ready to either touch, grasp top, or grasp bottom. Okay, I might have oversold this as exciting and calling you predictable and all that, but you are predictable. This is a prediction about the way that behaviors will occur after the planning step that is normally totally invisible. So that's the big thing here. Using just exclusively this neuroimaging information, they can predict different hand movements before they actually occur, right? So it identifies this span of motor planning, and it demonstrates that, the brain is producing these unique signals and different patterns of activation, i.e. it's doing something different with the information that it has available to it, and shows that those are distinct enough to be able to call out the outcome. Now, in this case, it was restricted to 3 exact outcomes. You might even call this task a micro-world, right? But, well, there's some distinctions there. But just hope you recognize the parallels, at least. This is a limited set of actions, and so that's what the task is designed to test. But it does so. It succeeds in being able to identify those actions before they are real, which is great. So that's another take on how we can use neuroimaging techniques to figure out what's going on inside the brain, and in the processor model of the brain. There's also some interesting recent work from the Gallant Lab at UC Berkeley, 2016. This one is cool. This is sort of an advancement in the sort of granularity of how we're trying to map what's going on in the brain. And they were able to chart this sort of map of semantics of different words all over the brain. So when you're thinking about bird And when you think about the verb that it does, fly, those are two different little chunks of the brain that are active but also connected. And this is the idea. So I'll let them do their thing, assuming they speak to me. So hang on, mute myself.
This is a map of someone's brain, showing roughly which areas respond when they hear different words. For example, there's a small area in the middle frontal gyrus that reliably responds to hearing the word top. But it's not just one word, one location. A single word can activate a whole range of different brain regions. So we find the word top in a bit of brain that seems to respond to words associated with clothing and appearances. But also here with numbers and measurements. And here with buildings and places. We usually think of language as being restricted to certain sections, like the temporal lobe. So researchers were surprised to find activity all across the brain and in both hemispheres. The map was made by scientists at the University of California, Berkeley. They put volunteers in an MRI scanner and had them listen to stories for 2 hours.
Back in the front again, deep, and she pulls out a pack of matches that have been laundered at least once.
By monitoring blood flow to different parts of the brain, they worked out which places were responding to the meaning of the words, the semantics. They found that different bits of the brain responded to different kinds of words and concepts. And they could group them into rough categories, shown here by the different colours. Dark green bits, for example, were most activated by words to do with numbers, red bits by social words. Here in the right temporoparietal junction, this speck of brain, just a few millimetres across, was found to respond to words like wife, mother, pregnant and family. And this bit just next door responds to some of the same social words like family and wife, but also words to do with places and people like house and owner. Generally, the concepts represented in each brain region relate to other functions that scientists already know about. So words to do with how things look, such as stripes, are likely to be found near the visual cortex. And although each individual's map is different, looking at these three brains, it's clear that different people have the same kinds of concepts in the same kinds of places. This is the first time we've been able to map the semantic systems of the brain in such detail, discovering that words are grouped by meaning, and revealing just how complicated and widespread the word maps in our heads really are. And the brain map is available online for anyone to explore.
Very interesting, right? Yeah, they've been at this for a while, and I believe this is a slightly more up-to-date version of the same kind of investigation of it's fascinating, right? We're seeing this evolution into this really high resolution spatial understanding of where even small concepts can live, and it's just great stuff. Active research. There's one other video to show from the same group to just show some interesting stuff about what they've got. Hang on.
Here in the functional MRI room at the University of California, Berkeley, it's story time. All in all, getting a brain scan for this project isn't a bad gig. Just kick back, watch some videos, listen to some stories.
From PRX, this is the Moth Radio Hour. So all the person has to do is lay there and listen to the stories and think about, the stories as anyone would.
Okay, Mark, we're going to start now. Are you ready?
But obviously, this is much more than a midday break.
Okay, great, thank you.
With support from the National Science Foundation, neuroscientist Jack Gallant and his team are using big data techniques to analyze these brain scans and map where in our heads we process language-related information.
So we can measure changes in blood flow throughout your entire brain simultaneously, about once a second. We have very large data sets, and we need to essentially drill down and mine into those data sets to recover the important functional information.
How's it going to grow a flower?
This visualization shows brain activity patterns that play out as a person listens to a story.
So on the right here is sort of a karaoke view of a story that people are listening to.
He made holes with the dibber and set the bulbs inside, and he was careful when he patted down the dirt so they wouldn't turn.
Colors show parts of the brain that are active at each point in the story.
Red tends to be parts of the stories that's representing meaning in the stories that is social. Green tends to be the material properties of objects and the structure of the world. So you can see now we're switching, and you see that the patterns of activity become much more green.
They estimate there are 200 to 400 modules spread all around the brain where functional thought occurs. 100 to 150 of those are linked to language processing. Red here shows the areas of more brain activity, blue areas of less.
So on the right here, we have some movies that subjects were shown in the magnet.
They're also using the fMRI data to build a mathematical model, or decoder, that accurately predicts how the brain extracts meaning from silent movies.
Here's an ocean scene. You can see it gets fish. Okay, it thinks there's a flower. That's not perfect, but you can see it gets water, swimming, ocean floor.
Gallant says practical applications could one day include new therapies to help stroke patients recover language skills, faster, more nimble computers, even brain-machine interfaces that would allow communication without speech.
In order to, heal people that have diseases or broken brains, or to apply the knowledge we get from neuroscience to, say, build better computers, we need to really understand the brain at a fundamental level.
That's not a job they take lying down. For Science Nation, I'm Miles O'Brien.
Ugh, Miles. Yuck. Last pun. Yeah, so again, in the same realm of building models that can, on the fly, take neural information and map it to a prediction about what that thing is. It's just all a little bit more internal. As opposed to physical actions, it's more of semantic activations. So, great stuff. So all in all, this is the whole idea of what we can do to map brain activity. And these neuroimaging techniques that we have come to possess as a society are pretty powerful, right? They've helped us really dig into the nature of how we process information and make sense of our world. And so the questions of, what do these different regions do? How do they toss information back and forth between each other? And how is that information coded and transformed? That one still has some work to be done, but that's the stuff. So that's today, and that takes us to our final thought. Think for a moment about learning the meaning of an ostensibly English word you've never seen before. Wargarble. What does it mean to learn its meaning? What other brain regions would need to be connected to it for it to make sense? Visual or auditory processing, action planning, the meanings of other words? All right. Thanks. Take care.